Welcome to your body's clues. When it comes to the brain, one of the most persistent questions is this: are male and female brains truly different? We've all heard the myths: men are more logical, women are more emotional, men have bigger brains, women connect both hemispheres better. But how much of this is cheap storytelling? And how much of it reflects the expensive truth uncovered by neuroscience? In this episode, we'll dive into the science of brain differences between the sexes, exploring what's real, what's exaggerated, and what these differences mean for health, behavior, and identity. There are, of course, biological differences between male and female brains. From the very beginning, even in the womb, sex hormones such as testosterone and estrogen influence neural development. Later in life, those same hormones continue to shape brain function, affecting mood, memory, and behavior. Evolution, too, plays its part. Natural selection has refined the human brain over millions of years, but it hasn't acted alone. Sexual selection has also guided the way, shaping traits related to attraction, reproduction, and competition. But here's where things get tricky. When we talk about sex differences in the brain, it's dangerously easy to slip into what biologists call just so stories, neat, oversimplified explanations that sound plausible but aren't necessarily backed by evidence. For example, the idea that men evolved to be better at navigation because they were hunters, while women evolved to be better at multitasking because they were gatherers. These stories are appealing, but they're often built on shaky scientific ground. This is exactly why neuroscientists working in this area have to fight so hard against popular myths. Some of the most common ones include the belief that men's larger brains automatically mean they are more intelligent, the stereotype that women are wired for empathy and men for logic, the assumption that cognitive abilities can be neatly divided into male and female categories. In reality, the differences that do exist are often subtle, overlapping, and far less dramatic than the myths suggest. Modern neuroscience shows us that while sex hormones and evolution do shape the brain, individual variation is far greater than group differences. In other words, two men may differ from each other more than an average man and woman differ. The challenge, then, is to separate cultural stereotypes from biological science, to recognize that while biology matters, it doesn't dictate destiny, and it certainly doesn't support the crude myths that have been passed down through history. One of the most persistent myths is that men are inherently smarter than women because, on average, they have larger brains. At first glance, the logic seems straightforward: bigger brain, more intelligence. But neuroscience tells us a very different story. Yes, men's brains are slightly larger on average, but that's mostly because men, on average, have larger bodies. Brain size scales with body size, and when you control for this factor, the difference largely disappears. What really matters isn't raw size, but how the brain is structured, how densely it's packed with neurons, and how efficiently those neurons communicate. Brains can vary enormously between individuals. A smaller brain may be more densely wired or have more efficient white matter connections, allowing it to process information faster than a larger one. In fact, many women have brains that are larger than those of many men, completely undermining the myth of a neat division between sexes. The relationship between brain size and intelligence is complex and only moderately correlated. Larger brains don't guarantee higher IQ scores, just as smaller brains don't mean lower intelligence. What counts is the quality of neural architecture, how well the networks are organized, how adaptable they are, and how experience sculpts them over time. This is why neuroscientists caution against equating size with ability. Bigger doesn't mean better when it comes to brains, and reducing intelligence to simple measurements is not only scientifically inaccurate but also socially harmful. It's true that scientists have found both structural, morphological, and functional differences in the average male and female brain. For example, men tend to have a slightly larger brain volume on average, while women often show greater cortical thickness in certain regions. Some studies also suggest subtle differences in how each sex tends to use brain networks, with women showing stronger interhemispheric connectivity and men showing slightly stronger intrahemispheric connections. But here's the crucial point: of variation within each sex is enormous. Two men can differ more from each other in brain structure and function than the average male brain differs from the average female brain. The same holds true for women. This means that while sex-based averages exist, they are often overshadowed by individual differences shaped by genetics, environment, experience, and even lifestyle. For instance, education, stress, nutrition, and social context can all alter brain wiring and function, sometimes in ways that dwarf any biological sex differences. So yes, male and female brains are not identical, but the idea that we can predict someone's intelligence, personality, or creativity simply by their sex is not supported by science. Brains are more unique than they are gendered. Each one is a product of biology, environment, and experience, a one-of-a-kind pattern rather than a simple blueprint of male or female. When neuroscientists compare large groups of men and women, clear average differences emerge. These may include variations in overall brain volume, connectivity patterns, or hormone-driven influences on certain regions. On a population level, such findings can be statistically reliable. But here's the catch: applying these averages to individuals is both misleading and scientifically unsound. The reason is simple: the overlap is enormous. For nearly every measurable trait, from brain size to cortical thickness to patterns of connectivity, the distributions for men and women overlap far more than they separate. Think of it this way: while men may have slightly larger brains on average, countless women have larger brains than many men. The same goes for functional traits: memory, spatial ability, emotional regulation all vary so widely from person to person that individual differences often outweigh sex differences entirely. What this means is that brains don't fit neatly into male and female categories. Instead, they exist on a spectrum of diversity, where each individual is a unique blend of traits. Morphology, wiring, and function are shaped not just by sex, but by genetics, development, experience, and environment. So while averages may tell us something about populations, they tell us very little about you as an individual. To assume otherwise is not just ill-informed, it reduces the incredible complexity of the brain to a stereotype. When we talk about average differences between the brains of men and women, it's crucial to remember what averages actually mean. They describe populations, not individuals. Any given person you meet may fall almost anywhere along the continuum of traits. For example, while men on average may have slightly larger overall brain volumes, there are countless women whose brains are larger than many men's. Similarly, while women often show stronger interhemispheric connectivity, many men share this pattern as well. These findings don't form to neat, separate categories of male brain and female brain. Instead, they reveal a broad spectrum of variation in which the sexes overlap far more than they differ. This overlap is so extensive that knowing someone's sex tells you surprisingly little about the actual details of their brain. Individual variability driven by genetics, environment, experience, and lifestyle often outweighs sex differences entirely. So, when we hear that men and women differ on average, the key takeaway should be humility. Averages can describe groups, but they don't define people. Every brain is unique, and it's this individuality, not broad stereotypes, that shapes intelligence, creativity, and personality. In fact, one of the most striking pieces of evidence against the male brain versus female brain idea came from a large-scale brain imaging study published in 2015. Researchers analyzed thousands of scans, looking at everything from the number of neurons in certain regions, gray matter, to the density of nerve fibers that connect those regions, white matter, to the overall architecture of brain connectivity. What they found was surprising. Most brains contain a mosaic of both male typical and female typical traits. Instead of falling neatly into one category or the other, individuals had unique blends, a little more like the average female pattern here, a little more like the average male pattern there. This means that very few people have a brain that is entirely male or entirely female. Instead, the vast majority of us carry a mixture of traits that defy simple labels. It's more accurate to think of each brain as a unique fingerprint, shaped by biology, development, and experience, rather than as belonging to one of two rigid categories. The implication is profound. The myth of a strict male-female brain divide oversimplifies reality. Our brains don't respect binary boundaries. They are diverse, flexible, and individualized, and that diversity is part of what makes human cognition so rich. Most brains are not neatly male or female. Instead, they are a mosaic, a blend of traits traditionally associated with both sexes. Structural features such as gray matter density, white matter tracks, or patterns of connectivity rarely line up in a way that allows a neuroscientist to confidently say, this is a male brain or this as a female brain, and the mosaic extends beyond anatomy into behavior. When the same researchers examined large data sets for stereotypically male or female behaviors, things like aggression, nurturing, risk-taking, or empathy, they found that individuals were just as mixed. In fact, only about 0.1% of people displayed exclusively male typical or female typical behaviors. That means nearly everyone carries a unique combination of traits, some leaning toward the traditional male average, some toward the female average, and many falling somewhere in between. The idea of a strictly gendered brain simply doesn't hold up under scientific scrutiny. What this tells us is that the brain resists binary labels, instead of two separate categories. What we see is a continuum of diversity, where every individual brain is its own distinctive pattern, a one-of-a-kind mix that defies stereotypes. And yet, we often come across studies claiming to show significant differences between male and female brains. These headlines can be confusing, especially when other research emphasizes just how much overlap exists. So how should we make sense of these seemingly conflicting findings? The first step is to recognize that not all differences are measured the same way. Scientists may look at absolute brain volume, connectivity between regions, hormone levels, or patterns of activation during a task, depending on which metric is chosen. The results and the interpretation can vary widely. Second, we need to ask whether the differences are statistically significant but practically small. A tiny shift in averages can be mathematically real, but it doesn't necessarily translate into meaningful behavioral or cognitive differences. For example, a measured difference of 2% in gray matter density might be reported as men's and women's brains differ, but in practice that variation could be well within the range of normal individual differences. Finally, context matters. Cultural influences, life experiences, and environmental factors can all shape the brain just as strongly, sometimes more strongly than biological sex, without careful interpretation. It's easy to mistake a population-level trend for a hardwired truth. So before we leap to conclusions, we need to agree on how differences are defined, measured, and interpreted. Only then can we understand what those differences actually mean and whether they matter at all in real life. In biology, when males and females of the same species show consistent physical differences, we call this sexual dimorphism. These differences can be subtle, like body size, or striking, like the elaborate tail feathers of the peacock compared to the plainer plumage of the pigeon. Sexual dimorphism exists across the animal kingdom. Male lions grow manes, while lionesses do not. Male deer sprout antlers, while females generally don't. In some species, The differences are reversed. Female anglerfish are many times larger than their tiny parasitic male counterparts. These physical differences often arise because of sexual selection. Traits that improve chances of reproduction get passed on, even if they aren't directly related to survival. A peacock's heavy, ornate tail might make it harder to escape predators, but if it increases his odds of attracting a mate, the trait persists and becomes exaggerated over generations. When it comes to humans, sexual dimorphism is far less dramatic, but it still exists. On average, men tend to be taller and have more muscle mass, while women tend to have higher body fat percentages. Traits linked to evolutionary pressures, hormones, and reproductive roles. But when we turn to the brain, the picture is far more complex. And like feathers or antlers, neural traits don't neatly split along sex lines. Instead, they form a mosaic of overlapping features, with individual differences often outweighing sex-based patterns. When neuroscientists search for sexual dimorphisms in the human brain, the results always need to be interpreted carefully. That's because context and variability matter just as much as sex. Take two individuals, a man and a woman who grew up in the same socioeconomic environment, share a cultural background, and are roughly the same age. When tested on cognitive tasks, these two people are likely to perform more similarly to each other than to men of different ages, education levels, or cultural contexts. In other words, factors such as education, environment, nutrition, stress, and cultural influences can create differences in brain function that are far greater than the differences typically attributed to biological sex. A person's life history shapes their brain just as powerfully as their genetics or hormones. This is why interpreting brain data is so complex. Yes, averages may reveal small structural or functional sex differences, but the overlap between individuals is massive. And the influence of non-biological variables often outweighs those differences entirely. 2. Reduce brains simply to male or female misses the bigger truth. Each brain is a unique product of biology, experience, and environment. When studies report morphological differences between male and female brains, it's important to look at how those studies were designed. The human brain shows enormous variability in both overall size and in the shape and function of specific regions. Two people of the same sex can differ dramatically in brain volume, cortical thickness, or connectivity, sometimes more than the average difference between men and women. This is why research in this area must rely on large sample sizes. If a study only includes a handful of participants, the results may be skewed by individual outliers. One man with a particularly small hippocampus or one woman with an unusually large frontal lobe could distort the averages in a small data set. By studying hundreds or even thousands of participants, neuroscientists can filter out this natural variation and detect patterns that are truly meaningful at the population level. Even then, those patterns tend to be subtle, overlapping, and far less dramatic than the myths suggest. So when we hear about brain differences between men and women, we should always ask, how big was the sample? How robust were the findings? And do they actually matter for behavior and cognition, or are they simply small statistical quirks amplified by headlines? When it comes to predicting differences in the brain, age may actually be a stronger factor than gender. Across the lifespan, the brain undergoes profound changes both in its structure and in how it functions. What's fascinating is that even the sexual dimorphisms we often hear about aren't fixed. They shift with time. Some differences that appear early in life tend to fade or attenuate with age. Others become more pronounced, influenced by hormonal shifts, life experiences, or even neurodegenerative processes. For example, certain regions associated with memory and executive function may shrink at different rates in men and women as they age. But these patterns aren't uniform and are influenced by a host of other factors like health, education, and stress levels. And here's another key point: morphology doesn't always equal function. Just because one region looks larger or smaller in one sex at a certain age doesn't mean that cognitive abilities or behaviors map neatly onto those differences. Brain volume, cortical thickness, and connectivity don't always translate directly into performance. This reminds us that the brain is a dynamic organ. It's shaped not just by sex, but by age, environment, experience, and countless other variables. Understanding differences means looking at the whole context, not just one snapshot in time, when we talk about gender differences in the brain. It's important to remember that the brain is not a static machine, it's a moving target. And like bones or organs that largely maintain their form, the brain is constantly reshaping itself in response to experience, environment, and learning. This property, known as neuroplasticity, is the very strength of our most complex organ. New connections are formed, old ones are pruned, and pathways are rewired throughout our lives. Neuroplasticity is the reason children can recover from brain injuries far more easily than adults, and why learning a new skill at any age literally changes the brain's structure and function. But plasticity is not limitless. It is shaped and constrained by biology, by genetics, hormones, and the natural limits of brain architecture. This means that while the brain is flexible, it doesn't completely erase the influences of biology or development. Instead, it creates a constantly shifting interplay between nature and experience. This variability adds another layer of complexity to the study of sex differences. A snapshot of a man's and a woman's brain at a single moment in time may reveal certain contrasts, but those contrasts are embedded in an organ that is continually adapting. What looks like a difference today may change tomorrow, depending on life stage, environment, or even personal habits. So when we ask how male and female brains differ, we're not asking about two fixed blueprints, we're asking about two dynamic systems, shaped by biology but sculpted by experience, ever-changing and never fully defined. When scientists study gender differences in the brain, it helps to sort them into three broad categories. This framework makes it easier to separate myths from meaningful findings. Always present and dimorphic, these are the clearest, most fundamental differences by definition. They are linked directly to sexual reproduction and sex hormones. For example, the neural circuits that control ovulation exist only in women, while those that regulate sperm production exist only in men. These dimorphisms are binary. They belong almost exclusively to one sex or the other, and they don't overlap. Distributed along a continuum, the second category includes traits where both sexes share the same feature, but their averages differ. Think of it like height. On average, men are taller than women, but there are many tall women and many short men. The same principle applies to certain aspects of brain size, connectivity, or even cognitive performance. These are statistical differences, not absolute divisions, changing over time. The third category reflects differences that are dynamic rather than fixed. Men and women may appear more distinct in a certain trait during one stage of life but converge later, or the reverse may happen. For instance, hormonal changes during puberty, pregnancy, or menopause can influence brain function in ways that alter sex differences across the lifespan. Similarly, age-related decline may shrink some gaps or expand others. This framework highlights why interpreting gender differences in the brain is so complex. Some differences are fixed, others are statistical tendencies, and still others shift with time, biology, and environment. Simplifying all of this into male brains versus female brains ignores the real picture. The brain is diverse, adaptable, and far more nuanced than stereotypes suggest. Mischaracterizing the biological differences between male and female brains is problematic enough, but there's an even more damaging mistake we often make: assuming that structural or functional brain differences automatically translate into behavioral differences. This leap of logic fuels stereotypes. If we say that women's brains are wired for empathy or that men's brains are built for logic, we begin to treat those generalizations as destiny. From there, it's a short step to discrimination, excluding people from opportunities, diminishing their achievements, or limiting their choices based on a mythical idea that gender can predict how someone thinks, feels, or behaves. The reality is that brain differences, where they exist, are statistical trends, not absolute truths. They describe populations, not individuals. Every brain is a unique mosaic of traits, shaped not only by sex, but also by age, culture, environment, and experience. To assume that sex alone can define behavior is to ignore this complexity. And yet, history is full of examples where such flawed reasoning has justified prejudice, from claiming that women are less capable of higher education, to suggesting that men are less suited to caregiving. Each time, the argument has leaned on a distorted reading of biology. This is why modern neuroscience insists on caution. Yes, sex matters in biology, but it doesn't dictate destiny. The most dangerous myth isn't just that men's and women's brains are different, it's that those differences can be used as a blueprint for how individuals should live, think, or be treated. There are indeed several key regions of the brain where average differences have been reported between heterosexual men and women, even after adjusting for overall body size. These include the amygdala. This almond-shaped structure is central to the emotional coloring of memories. It helps decide what experiences are worth remembering and links emotions like fear, pleasure, or stress to those memories. On average, men tend to show slightly larger amygdala volumes, but the functional implications of this are far from straightforward. The hippocampus, known as the brain's memory architect. The hippocampus is essential for forming new long-term memories and navigating through physical and mental space. Some studies suggest women may have proportionally larger hippocampi, potentially reflecting differences in spatial navigation strategies and memory-related processing. The corpus callosum, this thick bundle of nerve fibers, is the highway of communication between the two hemispheres of the brain. Earlier research suggested that women might have proportionally larger or more robust corpus callosa, fueling theories that women integrate information across hemispheres more efficiently. Later studies, however, have shown that the picture is much more nuanced, with differences depending on the specific region measured and the methodology used. But here's the catch: the story isn't nearly as simple as men have this, women have that. Differences are not absolute, and they don't map neatly onto behaviors. A larger hippocampus doesn't automatically mean better memory, and a larger amygdala doesn't automatically mean stronger emotions. Moreover, lifestyle, culture, stress, and experience can reshape these brain regions in powerful ways, often blurring or even reversing the sex-based averages. This is why neuroscientists stress caution. While these differences exist statistically at the population level, they don't define individual abilities or destinies, just as two people of the same sex can differ dramatically in these regions. Many men and women overlap completely, showing that brain diversity is far richer than a simple male-female divide. On average, the amygdala tends to be slightly larger in men than in women. This almond-shaped structure, buried deep within the temporal lobes, plays a critical role in how we process and remember emotional experiences. One of its key jobs is to act as a kind of emotional amplifier for memory. When something highly emotional happens, whether it's terrifying, joyful, or deeply moving, the amygdala sends a signal to the hippocampus, essentially saying, "Pay attention, this matters. Store this more carefully." This is why we often remember the details of emotionally charged events, such as where we were during a major life moment, far more vividly than we remember the details of an ordinary day. The amygdala prioritizes what feels important for survival or significance. But here's where it gets interesting. The size of the amygdala doesn't directly predict emotional sensitivity or memory strength. Function depends on many factors such as hormones, stress levels, life experience, and even cultural expectations. For example, women often report stronger emotional recall than men, despite having smaller amygdalae on average, suggesting that connectivity, hormone influences, and socialization may matter more than raw size. So while the amygdala is larger in men, its role is not about gendered emotion but about the universal human process of tagging memories with emotional W. 8. It serves as a reminder that size alone rarely tells the full story of how our brains shape thought and behavior. Studies that look at how men and women recall emotional events reveal an interesting pattern. On average, women tend to have stronger and more detailed recollections of emotionally charged experiences. Not only are the memories richer in detail, but women also seem to bring them to mind more quickly. This means that the emotional boost to memory, that extra push from the amygdala telling the hippocampus to pay attention, is generally larger in women than in men. The result is that women may recall where they were, what they felt, and even small contextual details of an emotional moment with greater precision. But this finding doesn't mean women are more emotional or men are less emotional. Instead, it likely reflects a combination of biological, hormonal, and social factors. Hormones like estrogen, which can influence memory circuits, may play a role. So might cultural factors. Women are often encouraged to pay closer attention to feelings and relationships, which could reinforce memory patterns over time. The key takeaway is that while sex differences in emotional memory exist on average, they are not absolute. Many men have vivid emotional memories, and many women do not. Once again, what we see is a statistical trend within overlapping populations, not a rigid rule. At first glance, the enhancement of emotional memory might sound like a gift. After all, remembering important details more vividly could help us learn from experience and navigate future challenges. But this strength can also carry a cost. Women, on average, are diagnosed more frequently than men with depression, anxiety disorders, and post-traumatic stress disorder (PTSD). One possible reason is that stronger emotional memory may also mean that painful or traumatic experiences are encoded more deeply and are harder to forget or move past. The very mechanism that makes joyful moments more vivid can also intensify the memory of grief, fear, or trauma. Interestingly, some studies suggest that while women tend to remember emotional events with greater clarity, their memory for the neutral moments just before those events may be weaker than men's. This could mean that surrounding context is sometimes lost when an emotional event dominates the memory system. Taken together, these findings highlight how neurobiology is rarely all benefit or all drawback. A trait that provides resilience in one setting can increase vulnerability in another. Emotional memory is not simply better in one sex, it's different, with trade-offs that shape mental health risks in complex ways. There is also compelling evidence that the amygdala plays different roles in men and women when it comes to sexual behavior. On average, the male amygdala is larger than the female amygdala even after adjusting for total brain size. This difference isn't random. The amygdala is densely packed with sex hormone receptors, making it especially sensitive to hormones like testosterone and estrogen. Because of this, the amygdala doesn't develop identically in men and women. Instead, its structure and function are shaped by the unique hormonal environments that the two sexes experience, beginning even before birth. For example, higher testosterone exposure in the developing male brain influences the way the amygdala grows and later responds to social and sexual cues. Meanwhile, in women, estrogen and progesterone shape the amygdala differently. leading to distinct patterns of reactivity and connectivity. Research suggests that these differences help explain variations in sexual behavior, attraction, and even emotional reactivity between men and women, but it's important to remember that these findings describe population-level averages, not individual destinies. Hormones interact with culture, personal experiences, and individual biology, making each person's brain and sexual behavior highly unique. So while the amygdala does develop differently in men and women, likely because of sex hormone activity, this is just one part of a much larger and more intricate picture. Biology sets the stage, but environment and experience continue to shape the performance throughout life. Adjacent to the amygdala lies another critical structure, the hippocampus. This region is often described as the brain's memory hub, responsible for forming new long-term memories, organizing them, and even helping us navigate through space. Several studies have reported that, on average, women tend to have larger hippocampi than men. At first glance, this might suggest that women have a biological advantage in certain forms of memory, but the story is much more complex. One factor appears to be aging. Research including a 2001 study found that the hippocampus tends to begin shrinking in men as early as their 30s, while in women this decline often occurs later. This difference could partly explain why women sometimes show better performance in memory tasks across midlife. Another layer of complexity is stress, which doesn't affect the hippocampus in men and women in the same way. Stress hormones such as cortisol can damage hippocampal neurons and impair memory, but studies have shown that men and women respond differently depending on the type, timing, and duration of stress. For example, in certain conditions, stress may suppress hippocampal activity more strongly in men, while in others it may alter connectivity or memory performance in women. It's also important to note that hippocampal size alone doesn't determine memory capacity, connectivity, neural efficiency, and hormonal context all influence how the hippocampus functions. A slightly larger hippocampus doesn't guarantee stronger memory, just as a smaller one doesn't automatically mean impairment. So while the hippocampus does show average sex-related differences, those differences are layered with factors like age, hormones, and stress. Once again, individual variability often outweighs the averages, making it impossible to reduce memory ability or hippocampal function to simple categories of male or female. While several smaller studies have suggested that women may, on average, have a larger hippocampus than men, more recent and rigorous research calls this into question. A large-scale meta-analysis published in Neuroimage in 2015 pulled data from many studies and concluded that there are no consistent differences in hippocampal volume between men and women. Why the discrepancy? One likely reason is sample size. Smaller studies are more vulnerable to statistical noise and outliers. If just a handful of participants fall at the extremes, say a few men with unusually small hippocampi or a few women with unusually large ones, the averages can appear misleading. By contrast, a meta-analysis with thousands of participants smooths out those outliers, giving us a more reliable picture. This doesn't mean the hippocampus isn't influenced by sex-related biology. It certainly is, but its size alone may not show a clear, reliable difference between men and women. What seems more likely is that other factors, age, hormones, stress levels, genetics, and environment interact with sex in complex ways, sometimes producing differences, sometimes not. The broader lesson here is that when we're talking about subtle brain differences, replication and scale matter. Isolated findings can spark intriguing theories, but it's only when we gather large, diverse data sets that we can separate genuine patterns from statistical noise. On average, women's brains tend to have a greater proportion of gray matter, the tissue that houses the neurons responsible for processing information. problem solving, and higher cognition. Men's brains, on the other hand, often show a higher percentage of white matter, which is composed of axons, the long, fiber-like projections that connect neurons and allow different regions of the brain to communicate. At first glance, this might suggest a neat division: women with more gray matter, emphasizing local processing and detailed analysis, and men with more white matter, emphasizing connectivity across regions. But the reality is far more complex. The story becomes particularly interesting when we consider the corpus callosum, the largest bundle of white matter in the brain, connecting the left and right hemispheres. For decades, researchers debated whether women had a proportionally larger corpus callosum than men, which led to popular claims that women integrate information between hemispheres more effectively. However, later studies showed inconsistent results, with some supporting differences, others finding none, and still others suggesting the differences depend on which subregion of the corpus callosum is measured. Even when differences are present, interpreting what they mean for cognition and behavior is a challenge. A larger or differently shaped corpus callosum doesn't automatically translate into better communication between hemispheres. It could reflect variations in connectivity patterns, efficiency, or even developmental influences like hormones or environment. Ultimately, while gray and white matter proportions and corpus callosum size may show population-level averages, the overlap between men and women is enormous. Individual variability often outweighs sex differences, reminding us again that brains cannot be neatly classified into male and female blueprints. The corpus callosum, the massive white matter bridge connecting the left and right hemispheres, is often cited as one of the brain regions showing sexual dimorphism. Early studies, beginning with a much-publicized 1982 report, found that women tended to have larger and more bulbous corpora callosa than men. This finding was quickly interpreted in popular media as evidence that women's brains had superior interhemispheric communication, allowing them to think more holistically, while men's brains were cast as more lateralized and specialized. It was a compelling story, but also an oversimplified one. Later research began to complicate the picture. Some studies replicated differences in specific subregions of the corpus callosum, while others found no meaningful differences at all once total brain size was factored in. Meta-analyses have suggested that many of the early results may have been skewed by small sample sizes, inconsistent measurement techniques, and publication bias, even if we accept that slight morphological differences exist. Interpreting them as direct evidence of holistic versus logical thinking is a leap too far. A larger corpus callosum doesn't necessarily mean better communication across hemispheres. It could reflect differences in how connectivity is organized, how efficient information transfer is, or even how development is shaped by hormones and environment. Modern neuroscience now sees the corpus callosum not as a marker of gendered cognition, but as one piece of a far more complex connectivity puzzle. Brain function emerges from vast networks of circuits that crisscross hemispheres and regions, making simple claims about women being holistic and men being logical less science and more stereotype. The scientific debate over sex differences in the corpus callosum shows just how complicated brain research can be and how easily early claims can snowball into myths. After the much-discussed 1982 study suggesting women had proportionally larger and more bulbous corpora callosa, a second paper in 1991 refined the claim. The female corpus callosum appeared more bulbous, while the male structure seemed more tubular, with the total area roughly the same. This nuanced finding undercut the idea of a simple, one-directional difference. By 1997, a meta-analysis covering 49 studies delivered an even stronger verdict across large samples and varied methodologies. There were no significant sex differences in corpus callosum size or shape. This was a major blow to the stereotype that women's brains are inherently more interhemispheric. Yet the story didn't end there. In 2003, a study of Indian brains introduced new complexity, reporting that the corpus callosum was longer in males than in females, and that it tended to increase with age in men but not in women. While intriguing, these findings highlighted another key issue: population differences matter. Results may vary across cultures, genetic backgrounds, and environments, complicating the search for universal truths. Taken together, the research paints a clear picture. While early studies fueled the idea that the corpus callosum reflects stark sexual dimorphism, the bulk of evidence suggests that any differences are subtle, inconsistent, and heavily shaped by methodology and sample diversity. What's more, even when small differences are observed, they don't neatly translate into predictable differences in cognition or behavior. The corpus callosum debate is a powerful reminder that in neuroscience, as in all science, replication and context are everything. One or two striking studies can spark cultural myths, but only large-scale, rigorous analysis can reveal the real story, and in this case, that story is far messier and far less gendered than popular accounts suggest. What these studies ultimately reveal is that measuring brain volumes is anything but straightforward. Even when researchers focus on the same region, whether it's the hippocampus, the amygdala, or the corpus callosum, the results can vary dramatically. One reason is variability with age. Brain structures don't remain static, they change across the lifespan, shrinking, thickening, or reorganizing in ways that can obscure sex-based differences. A study showing a difference in younger adults may not hold true in older populations, and vice versa. Another factor is cultural and population diversity. Brain morphology can differ across ethnic and cultural groups, as seen in the 2003 study of Indian brains, suggesting that local genetics, diet, health, and even environmental pressures influence anatomy in ways that complicate universal claims. And, finally, there are methodological differences: how regions are defined, which imaging techniques are used, and how data are analyzed all matter. Small changes in measurement can yield very different results. The take-home message is clear: while sex differences in brain structure are real in some contexts, they are far from simple or universal. Variability across individuals, lifespans, and populations often muddies the waters so much that any neat, one-size-fits-all explanation collapses under scrutiny. Some of the research on brain connectivity has been misused to support the familiar but misleading left-right brain myth. According to this narrative, women's brains supposedly have stronger connections between the hemispheres, which gives them greater access to the so-called intuitive, emotional right hemisphere. Men, on the other hand, are said to be dominated by the logical, analytical left hemisphere. It's an idea that sounds appealingly simple, but it distorts the science. While some studies have found subtle differences in how men's and women's brains are wired, men often showing more connections within hemispheres, and women showing more cross-hemisphere links, the leap from connectivity patterns to sweeping claims about intuition or logic is unsupported. For one, the right hemisphere is not solely emotional and creative, nor is the left hemisphere exclusively logical and analytical. Both hemispheres participate in nearly every cognitive task, from problem-solving to emotional processing. Communication across hemispheres is constant, and it happens in men's brains just as much as in women's. Furthermore, the differences in connectivity are statistical averages, not rigid categories. Individual men and women show enormous overlap, and cultural, educational, and experiential factors often play as much of a role as biology. The persistence of the left-right brain myth shows how easily nuanced neuroscience can be flattened into stereotypes. The reality is far more fascinating. Our brains work as integrated networks, not dueling halves, and both men and women draw on the full richness of those networks every day. There is still a pressing need for more rigorous research on sex differences in the brain. While much of the popular conversation is dominated by oversimplified myths, the truth is that real, measurable differences, when they exist, carry important implications for health and medicine. For example, we know that the prevalence of many psychiatric and neurological disorders differs between men and women. Depression and anxiety are more frequently diagnosed in women, while disorders such as autism spectrum conditions and ADHD are more common in men. Neurodegenerative diseases like Alzheimer's also show different patterns of risk and progression between the sexes. To properly diagnose, prevent, and treat these conditions, we need to understand how male and female brains may differ at the levels of structure, function, and chemistry. If we ignore these differences, we risk applying one-size-fits-all treatments that fail half the population or worse, treatments that help one group but harm the other. At the same time, it's important to conduct this research carefully, avoiding the trap of exaggerating small effects into sweeping stereotypes. The goal isn't to reinforce myths about gender and ability, it's to build a more precise science of neurobiology that takes sex differences seriously while recognizing the immense overlap and individuality that characterize all human brains. In other words, studying these differences is not about dividing us into categories, but about improving health outcomes for everyone. Men and women may be exposed to the same conditions: stress, infection, trauma, or even a pharmaceutical treatment, yet their bodies and brains don't always respond in the same way. These differences can manifest in biology, physiology, and behavior, leading to diverging outcomes. For example, sex differences can be seen in disease prevalence. Depression, anxiety, and autoimmune disorders are diagnosed more often in women, while autism spectrum disorders, Parkinson's disease, and ADHD are more common in men. Prognosis: some conditions progress more rapidly or with different symptoms depending on sex. Alzheimer's disease, for instance, shows distinct patterns of decline between men and women. Response to treatment and medications that work well for one sex may be less effective or carry stronger side effects for the other. This can stem from differences in hormones, metabolism, or even how drugs cross the blood-brain barrier. Recognizing these differences is not about reinforcing stereotypes but about practical medicine. If researchers and doctors fail to account for sex, treatments risk being incomplete, imprecise, or even harmful. Understanding male and female neurobiology more deeply could lead to better diagnostics, personalized therapies, and improved outcomes for everyone. In short, acknowledging sex differences in the brain is not a matter of ideology, it's a matter of health, accuracy, and saving lives. Despite the clear importance of studying sex differences in the brain, there has been a long-standing bias in medicine and research toward focusing on male subjects. This is true not only for human patients but also for laboratory animals used in experiments. Among animal researchers, the justification has often been convenience. Male animals are considered easier to work with because they do not experience the estrous cycle, the recurring hormonal fluctuations in females. The argument goes that unless there is clear evidence of sex differences, it's simpler to eliminate the complicating variable of hormonal changes, but this approach comes at a cost. Hormonal cycles do affect behavior, cognition, and physiology, and ignoring them means we risk missing key insights into how female biology responds differently under certain conditions. For example, stress, learning, and even responses to medications can vary across the phases of the estrous cycle. By excluding females, researchers build results that may not fully translate to half the population. This male-centric bias has carried over into human medicine as well. Historically, clinical trials were disproportionately conducted on men, with the assumption that findings could simply be applied to women. Today, we know this is not only scientifically flawed but potentially dangerous, as it obscures differences in disease prevalence, symptom expression, and treatment response between men and women. Correcting this bias requires a shift in mindset. Female biology is not a confounding variable to be controlled away, but a vital dimension of understanding health and disease. Only by including both sexes in research can we ensure that medical discoveries are accurate, safe, and beneficial for everyone. Across decades of biomedical research, females have been consistently underrepresented both in animal studies and in clinical trials with human participants. This imbalance is more than a statistical oversight. It creates serious risks for women's health. Medications are not one-size-fits-all. Side effects, absorption rates, and effective dosages can vary significantly between men and women due to differences in body composition, hormone levels, metabolism, and even genetic expression. For example, dosage sensitivity. Women may require lower or different doses of certain drugs, such as sleeping pills, because their bodies metabolize the compounds more slowly. Side effects. Some cardiovascular drugs or antidepressants can cause disproportionately stronger side effects in women, which often went unnoticed until after approval. Efficacy. Painkillers, psychiatric medications, and even vaccines sometimes show differing levels of effectiveness between sexes, meaning that clinical results drawn mainly from men may not apply equally to women. Excluding women or treating them as simply smaller men ignores these differences. The estrus, or menstrual cycle, often seen as a confounding variable, is in fact a crucial biological factor influencing drug responses and brain function. By not studying it, researchers miss important insights that could improve treatments for both sexes. In short, women's underrepresentation in research has led to blind spots in modern medicine. Correcting this imbalance is not only about fairness but about safety, precision, and effectiveness in how we design drugs, test therapies, and treat patients. The effects of aging on brain volumes are not uniform across men and women. Research suggests that, on average, men experience a greater overall loss of brain volume as they age compared to women. This decline is often described as a more global shrinkage, affecting multiple regions of the male brain more evenly. Women, however, show a different pattern. Rather than losing brain volume evenly, they tend to experience region-specific reductions. For example, studies have found that the parietal lobe's areas crucial for spatial reasoning, sensory integration, and attention tend to decline more in women than in men. This means that while men may face broader reductions in brain mass, women may see sharper losses in functions tied to specific cognitive skills. These sex-based differences in aging trajectories could help explain why men and women sometimes show different vulnerabilities to neurological conditions later in life. For instance, men may be at higher risk for certain forms of mild cognitive impairment, while women's greater parietal lobe decline may contribute to the higher prevalence of Alzheimer's disease in women. It's important to note, however, that these are population-level averages. Individual variability is immense, and lifestyle, genetics, hormones, and health factors such as cardiovascular fitness, stress, and diet all influence the rate and pattern of brain aging. In other words, while sex matters, it is only one piece of the puzzle in how brains change with age. There is a compelling and urgent case for correcting the gender imbalance in medical research. For too long, women's biology has been treated as an afterthought, with medical studies defaulting to male participants and male animal models. This has created blind spots that limit the effectiveness and sometimes even the safety of treatments. If women are consistently excluded, then the medicines, therapies, and diagnostic criteria we develop risk being optimized for male bodies only. That means half the population receives care that may be less effective, less precise, or even harmful. By designing research that fully includes women, we can ensure that treatments are tailored to both sexes, not by erasing differences, but by acknowledging them. This isn't just a matter of fairness or representation, it's a matter of better science, safer medicine, and healthier lives. In short, correcting this imbalance is about ensuring that women benefit from medical breakthroughs equally, and that medicine lives up to its promise of serving all of humanity. Even the seemingly simple act of assigning gender in brain research is far more complicated than it might appear. Traditional studies often assume a straightforward binary, male and female, defined by anatomy at birth, but human reality is richer and more nuanced. Some findings about sex differences in the brain may not hold true for individuals whose gender identity does not align with their biological sex. Likewise, people who are non-binary, transgender, or whose sexual orientations are not exclusively heterosexual may present patterns of brain organization and function that don't fit neatly into conventional categories. For example, studies of transgender individuals have found that certain brain structures and connectivity patterns sometimes align more closely with their experienced gender identity rather than with their anatomical sex. Similarly, research into sexual orientation has shown variation in the size or activity of specific brain regions, suggesting that identity, biology, and behavior are intertwined in ways we're only beginning to understand. This underscores an important point. Our brains resist easy classification. While averages and patterns may help us understand trends, they should never be used to erase individuality. Each brain carries a unique combination of biological influences, lived experience, culture, and identity, making it a mosaic rather than a fixed blueprint. Ultimately, studying the interplay of sex, gender, and identity in neuroscience isn't just about science, it's about ensuring that the knowledge we build is inclusive, accurate, and reflective of the diversity of human experience. As we've seen, the story of male and female brains is not black and white. Yes, there are differences sometimes in size, connectivity, or hormone influence, but there is also massive overlap. Most brains are mosaics, blending traits that defy simple labels. The real danger isn't in studying differences, it's in misinterpreting them to reinforce stereotypes or justify prejudice. Understanding sex and gender in the brain is valuable not because it divides us, but because it helps us deliver better science, better medicine, and better care for everyone. So the next time you hear someone say, men's brains are this, women's brains are that, remember, the truth is more complex, more fascinating, and far less cheap than the myth. Thanks for watching Your Body's Clues. If this video opened your perspective, share it with someone curious about the mind. And don't forget to subscribe for more deep dives into how your body really works. For those who want to explore the science of sex differences in the brain more deeply, there are several key works worth highlighting. Coffey's Sex Differences in Human Brain Aging examines how male and female brains age differently, with distinct patterns of structural and functional change over time. Joel Berman Tavor, Wexler Gaber, and colleagues challenge the binary idea of male and female brains in their influential paper Sex Beyond the Genitalia, showing how most human brains are mosaics of traits traditionally labeled as masculine or feminine. McCarthy, Arnold, Ball, Blaustein, and DeVries provide an authoritative overview in sex differences in the brain, exploring the biological, hormonal, and developmental factors that shape neural variation between the sexes. Murphy and collaborators take a closer look at both structure and function in sex differences in human brain morphometry and metabolism, revealing how these differences may influence mental health and disease. Finally, Rugrock, Salini Khorshidi, Lai Baron Cohen, and their team bring together data from dozens of studies in a meta-analysis of sex differences in human brain structure, offering one of the most comprehensive and balanced assessments of where true differences exist and where they do not. Together, these works offer a nuanced and evidence-based view, moving far beyond myths to reveal the real and often surprising science of sex differences in the brain.